This video will go through DNA profiling in subtopic 3.5, genetic modification and biotechnology. In this video, the specific application that we'll talk about is the use of DNA profiling in paternity and forensic investigations. And the skill that this will allow is the analysis of examples of DNA profiles. We'll start with just recalling a few items. So when DNA is obtained, it is going to be subjected to that technique of PCR, and PCR is going to be used to amplify small amounts of DNA. That polymerase chain reaction is used to make large numbers of copies of DNA, and when undergoing PCR, that PCR cycle is going to repeat itself 25 to 35 times in a typical reaction, which will generally take 2 to 4 hours depending on the length of DNA region being copied. If the reaction is efficient, i.e. it works well, the target region can go from just one or a few copies to billions of copies. And the steps to PCR were covered in 2.7. So please see pages 155 to 156 of your textbook. Now, another thing to recall is that PCR does rely on a thermostable DNA polymerase, that Taq polymerase, and requires DNA primers designed specifically for the DNA region of interest. In PCR, the reaction is repeatedly cycled through a series of temperature changes, which will allow many copies of the target region to be produced. After PCR, the DNA sample is then subjected to gel electrophoresis. So gel electrophoresis is then going to be used to separate the fragments of DNA according to their size. So it will involve the separation of charged molecules such as DNA or RNA in an electric field according to their charge and size, but it'll be mostly due to their size. Interpretation of gel electrophoresis is done by comparing the unknown sample to a DNA ladder loaded into one of the wells. A DNA ladder is a sample of known length and sequence. So in gel electrophoresis, you're going to take your samples, apply them to the gel. An electric field is created that will cause the DNA fragments to migrate through the gel. And then just the longer molecules are going to migrate less in the gel, so they're going to stay closer to the top. Shorter molecules will migrate further in the gel, as they will be found closer to the bottom of the gel. Now, once that gel electrophoresis is complete, you will get a banding pattern that can be then analyzed. So in the applications of PCR, PCR can be used for DNA profiling. It can be used to amplify DNA found at crime scenes and in paternity cases. PCR is used to amplify genes associated with genetic disorders from the DNA of patients or from fetal DNA in the case of prenatal testing. PCR can be used to test for a bacterium or DNA virus in a patient's body. If the pathogen is present, it may be possible to amplify regions of its DNA from a blood or tissue sample. It can also be used to test for the presence of genetically modified food ingredients by using a primer that binds only to genetically modified DNA sequences. With DNA profiling now, DNA profiling will involve the comparison of DNA of different samples and to DNA found at a crime scene. So DNA from an individual or another source, such as a fossil or crime scene, is obtained. The desired sequence of DNA is selected and copied by PCR. The copied DNA is split into small fragments by restriction endonucleases, and you will need to review restriction endonucleases from the previous video. The fragments are separated using gel electrophoresis, and the banding pattern produced in the gel is then compared. So in the following example, we have our victim, the DNA sample obtained from the victim of the crime scene. Here we have blood collected from the crime scene. So here is our DNA evidence. These are subjected to PCR and then gel electrophoresis. Samples are collected from 3 suspects. Their DNA is then subjected to PCR and gel electrophoresis, and the banding pattern is compared from each suspect to that of the crime scene to determine which suspect most likely committed this crime. So when we compare the banding pattern, for example, in suspect number 1, comparing this to the DNA found at the crime scene, we can see this rather large band here in suspect 1 that is not present in the crime scene. Suspect 1 also has a shorter band that appears rather thick that is not present at the crime scene as well. Suspect number 3 has that similar banding pattern and has an even smaller fragment that's not found within that crime scene. Now, suspect number 2 has these two large fragments that appears to be similar to DNA found at the crime scene. It has this rather thick band of mid-size that's also found at the crime scene, and these two smaller fragments match DNA found at the crime scene as well. So in comparing the banding pattern of suspect number 2 to the banding pattern of the DNA found at the crime scene, we can say that suspect number 2 is the criminal here in this particular crime. DNA profiling can be used in paternity and forensic investigations. So DNA profiling can be used for bloodstains found in victims or at crime scenes, here found on victims or at crime scenes, semen from sexual crimes, or paternity cases where men claim they are not the father to avoid child support payments, or women who have had multiple partners may wish to identify the father, or a child may wish to prove that a deceased man was their father. So in this case here, when looking at this particular DNA profile, so here we have a mother who has had a child, and then there are 3 potential fathers here of this child. So in comparing the DNA from the child and the banding pattern that's produced by gel electrophoresis, looking at the potential fathers here, when we look at dad number 3 has these 2 small fragments of DNA, which are also found in the banding pattern of the child. Dad number 3 also has a larger DNA or a larger fragment that's also found in the DNA banding pattern of the child. We do not see these similar banding patterns from dad 1 or dad 2. So in this case, dad 3 is the most likely father of this child by using this technique of DNA profiling. So this is the end of this video for DNA profiling from subtopic 3.5.